Hi, good afternoon, everyone. Thank you very much for attending our December Core Technologies and Application Seminar Series. Today, we're excited to have Peter Faull who is the Director of the Proteomics Core Facility and a research assistant professor of biochemistry and molecular genetics. So we're really excited to have Dr. Faull here to talk about what they're doing in the proteomics core and how they work with the Cancer Center. So thank you.
Well, thank you very much. Good afternoon, everyone. As Sarah said, I'm Peter Faull. It's nice. I recognize some faces in the audience. So for those of you that I know, hello. Those of you I don't know, I look forward to meeting you. So just a little overview of what I'm going to talk about today. So I'm going to introduce myself, the staff on my team, and our core. I'm going to talk to you a little bit about what we actually do in terms of mass spectrometry-based proteomics and the services that we provide. And then I'll summarize, give an opportunity for some questions. But during this presentation, if you have any questions, just put your hand up and I'll try and address them right away. So to start with a couple of things. So introduction to me. You may recognize my accent is not from Chicago. I am from Northern Ireland, so I'm from near Belfast. I went to grad school in Scotland, University of Edinburgh, and I did a PhD in biophysical chemistry. I consider myself a chemist, and I explored how to measure the shape of proteins in the gas phase using a technique called ion mobility mass spectrometry. This was quite a long time ago, and nowadays ion mobility is a technique that's used to great effect within proteomics and other mass spectrometry techniques. And since 2012, so between my PhD, I did a postdoc in private industry, and then I've been working in proteomics cores basically since 2012. I started at the Medical Research Council in London. Then I did a stint at the Francis Crick Institute, also in London. During COVID, my husband and I moved over to Texas. I was at the University of Texas at Austin. They have an excellent proteomics core there. And then the summer before last, 2023, I was recruited to here at Northwestern. And I started in November of last year. So I just had my 1-year anniversary birthday here at Northwestern. So if you don't recognize me, but you've worked with the core before, it's because I've been here for just over a year. So that's a reason to come and talk to me. Clearly, the work that we do in the core is not all done by me. Actually, it's only a tiny part. It's this set of 5 incredible staff. And I always like to start with the staff. None of them are here today. They're all very busy. The 2 people at the top, Monica and Uday, they lead one of my services that involves histone analysis. And I'll talk about all these services later on. But if you've done any histone analysis or chromatin analysis, Monica and Uday might have worked with. In the bottom left, we have Rohan and Jyoti. They're my bottom-up proteomics experts. Protein ID, protein quant, they're the guys you might have dealt with. And then most recently, Dr. Simon Phillips, I recruited in September, just 3 months ago. He's our bioinformatics specialist. He actually worked in the core back in 2019. He left to go and get some experience again in private research, in some startup research. Might be a good person if you're considering after whatever field of study you're doing now. You're thinking of moving into the startup world in biotech, Simon's another great resource. Go and take him for a coffee and hear all about his work. But between us, this is the dream team. So really great. So the core's mission, and all the cores at Northwestern, we have our mission statements, whether it's to develop new proteomic technologies, to apply them to questions in basic, translational, and clinical research. Like today, it's about educating researchers throughout the Chicagoland areas. We work with UIC, University of Chicago, Loyola, Northwestern. And it's to communicate our research findings and your research findings to the wider scientific community. So the core was founded back in 2012, so it's over a decade old. It was initially up on the Evanston campus. And a question I get every week is, where are you guys? People think we're in Evanston. We're not. We're downtown. We came in September 2014, just over 10 years ago. It was moved downtown. We're next door in Folsom on the 8th floor. You're all more than welcome to come visit anytime. And the reason it was moved downtown is because the vast majority of people submitting samples in 2012 and 2013 were coming from the downtown campus. So rather than shuttling samples back and forth, it was decided there was space found and it was brought downtown, which makes a lot more sense. And so as of December 2024, We are really lucky to have just over 2,000 square foot of space, most of it in Ulsan. We actually have a little space up on the 7th floor of Lurie. We have 5 dedicated instruments, all state of the art. I'll talk about them later. 5 full-time staff, we just introduced, and myself. So today is not really going to be an in-depth lecture about mass spectrometry-based proteomics. I can give you all, email me, come and talk to me. I will talk for days about mass spectrometry and the different techniques. I can point you in the direction of great resources, great YouTube videos, primers, everything you want specifically for what you're interested in. But that's not going to be what today is about. I don't want to bore you with that. But let's just talk about some of the things that why we do proteomics and multi-omics in general. So going back to central dogma of molecular biology, this audience is probably very well versed in this. So our double-stranded DNA can undergo replication. Through the action of transcription, you form your single-stranded RNA molecule. Through translation, you form proteins. And from there, there's actually a further modification. So post-translational modification. In this example, I put a little P in a circle to indicate a phosphorylation. So the dynamics involved between forming proteins, a single protein, and then the number of additional jobs or roles it can have by being ultimately modified is why I refer to these frequently as the functional gene products. But again, as the world of multi-omics sort of increases, people doing transcriptomics or whatever, it really is just a huge opportunity to use the different cores and resources at Northwestern to use all of these in your research. And so just to take an example of so maybe a human genome, you're looking at maybe something in excess of 20,000 genes, the action of alternative promoters, alternative splicing, mRNA editing, you might get towards a transcriptome in excess of 100,000 transcripts. But after you do your post-translational modifications, you're easily in excess within the proteome of a million proteins. And so just that increasing sort of exponential complexity going from genomics through transcriptomics to proteomics. It's amazing that we can actually do this type of analysis to begin with. And so, brief introduction of mass spectrometry. So for those, I imagine raise of hands who's actually done some mass spectrometry in their research before? Yeah, a couple of people. So it's an analytical technique. It's used to measure the mass to charge ratio. That's the unit that we're actually detecting. It's frequently just referred to as M/Z or M/Z, and it requires gas phase ions. It's been around for a long time. It was pioneered late 19th, early 20th centuries. It was first called a mass spectrograph. It evolved into the mass spectroscope, and we're now in the world of mass spectrometers. And that's why the field is mostly called mass spectrometry, but you may read manuscripts or other areas that call it mass spectroscopy, but it's mass spectrometry. And so regardless of the type of mass spectrometry you're working with, all mass spectrometers, I think it's right in saying, is composed of 3 components. You have an ion source for generating your ions. You have a mass analyzer, which separates your ions, and then you have a detector to detect your ions. So it's really very simple, but it comes in a huge variety of instrumentation and flavors. And so we are very fortunate through the support of Northwestern, Lurie Cancer Center, and a number of grants and awards to have this array of state-of-the-art mass spectrometers. And I wanted to introduce them to you just so that if you're reading particular papers or manuscripts or publications, and today I'd like you to sort of take away some buzzwords or some jargon specific to proteomics, that if you hear it or see it again, you're like, oh, we have that capability here at Northwestern. Or we don't have that capability, but I maybe can help you find that capability somewhere, either through maybe we can get a grant to put it together, or one of my colleagues at another institute nearby can help you. So on the left of the screen, these 2 instruments are called Orbitrap Explorers instruments. And these are the instruments we use primarily for doing our bottom-up proteomics. That's our protein identification, our protein quantification. And within our research group and a number of other mass spec groups at Northwestern, led by Professor Neil Kelleher, we have a very cute way of referring to our instruments because they all have quite clunky names, quite long names, or we duplicate instruments. They all are named in a particular way. And you'll see that if you do work with us, when you get your results email from us, it'll state which instrument it was acquired on. And so you'll be able to see, so we have Lucy and Eve. Eve actually replaced an older instrument when I first arrived that was called Wall-E. And if you remember from the Wall-E movie, the girlfriend was Eve, slightly better, slightly more upgraded. So I thought that was kind of fun. In the middle, we have what's called an Orbitrap tribrid, and the tri is for triple hybrid, which means it's 3 mass spectrometers effectively in one. It's a quadrupole ion trap Orbitrap. So this instrument is really state of the art. We actually share it with a fantastic research group based in Ward, building next door of Jeff Sabas. And this instrument really is the best of the best that you can get. And again, these 3 instruments, the middle one and the 2 on the left, are for our bottom-up proteomics work. And so it's actually effectively my discretion. You come to talk to me, which instrument we're going to use is appropriate for your samples, your projects. And so we have some really lovely high complexity projects running on this instrument. And then finally, on the right-hand side, we are very fortunate to actually have 2 instruments that are effectively identical. These are TSQ Altus. These are triple quadrupole instruments. They're not quite as complex as the other 2, but they do a very dedicated type of analysis. And that's part of our histone identification and quantification workflows. And they're called Meg and Hercules, which I think are really super names. So again, you're looking at sort of $200,000-$300,000 plus, and then for the middle instrument, you're looking at closer to $2 million. These are basically Formula One race cars when it comes to analytical equipment. They're fantastic. So delving a little bit into what are the type of proteomic services we actually provide, before we talk about specifics, let me just make it clear, like all of the cores that you have access to here at Northwestern and within the Lurie Cancer Center, it's basically an end-to-end service. Use us as another additional tool in your research that you're doing. So come and talk to us. We want to talk to you, do a consultation prior to any of your projects. And this is so we can get to know you a bit better, your research a bit better, but also to help you avoid any of the common pitfalls, maybe give you some guidance on realistic aspects, a bit of a reality check about what is and isn't applicable. It's a great opportunity to talk to people about potentially how many replicates might be involved, or whether we just need to do a small pilot scale experiment. But fundamentally, the 3 things that we do for everybody is sample preparation, data acquisition, and data analysis. So for sample preparation, we will help guide you and tell you exactly what you need to do so as to maximize getting good results. And so I'll talk a little bit later about how we do that, but we have available to you so many protocols, so much resources and knowledge, we are more than happy to share. Data acquisition, obviously we do that on our mass spectrometers. There are a couple of research groups here at Northwestern that have their own dedicated mass spectrometers. So I put that there just in case if you're working with any of them, you can work with our instruments and their instruments. And then finally with data analysis, we have some wonderful pipelines for getting you really good, accurate protein ID and quantification information. And with the arrival of Simon Phillip in September, we have some really cool and interesting combinations of R scripting and pipeline generation data that we can do. But ultimately, you can choose to do your own sample preparation with our guidance. That might save you a little bit of time, maybe a little bit of money. If you want us to give you the raw data from the mass spectrometer, you can do your own data analysis as well. Maybe you have a collaborator or a pipeline already set up that's available, and we can help you with that. So that's other ways, but we will always do the data acquisition for you. And then after everything is done, we are more than happy follow-up discussions, data interpretation, to assist in manuscript writing. And something that is becoming much more important, prevalent over the last couple of years is that depending on where you are submitting your manuscript, your abstracts to, they may require raw data generated from omic technology to be uploaded to a public repository, something like Pride or Massive, and we can help do that. It's not particularly complicated, it just takes quite a lot of time. And also we have all of the raw data centrally located on a server. We can do it a lot quicker. For any of you that are applying for grants, applications, awards, I'm more than happy to give you a letter of support for that. And also then just to help guide next steps, next experiments, any of those things, please just reach out. We're here to help you guys. Okay. The 3 main services that we provide are shown here. we have bottom-up proteomics. We have our epiproteomic histone modification panel. It's a bit of a mouthful. I'll just refer to that as our histone analysis. And then also we have our top-down proteomics work that we do in collaboration with the Kelleher Research Group up in Evanston. So I'm going to talk through each one of these. I'm going to spend quite a bit of time on the bottom-up side because it's just sort of one flavor of mass spec. There's so many different subcontexts for that. Talk a little bit of histone and finish with the top-down work. Just to give some clarity, when I'm talking about these top-down, middle-down, bottom-up, what do they mean? So top-down proteomics refers to proteins that are relatively intact. They've not been disturbed. They may be native in nature, and you haven't done too many changes to them. That's a really challenging environment to do, and that's what we do in collaboration with the Kelleher Research Group. You then take those protein molecules and sort of digest them a little bit or just make them a little smaller, make them a little less complex. You move into a world called middle down, which is still quite a developing area. And it's an area I'd like to see more people come and talk to us about. It's not applicable to all research areas. It's something you'll see starting to gain more traction in the coming years. But most importantly, bottom up is where we take proteins that you guys provide to us, we digest them to peptides. So you've got quite a lot of heterogeneity, and then you work from the bottom up. You're using the mass spectrometer to identify and quantify the peptides. You're stitching that information back together using bioinformatic approaches, therefore to get your protein identification modification. And so I'm going to walk through a protein identification process. I'm going to take each of these steps in turn. This is quite a busy slide. But I think this sometimes helps give context to the number of steps that can be involved for a simple protein ID experiment. So the first step very much is sample preparation. So that's taking cells or tissue that you are working with in your lab and then ultimately producing peptides that we're going to inject into the mass spectrometer. And so we do that in a number of ways. Starting on the left-hand side, you need to start with cell lysis. You actually need to get the proteins out of the cells themselves or out of the tissue. And there's a huge number of ways that can do that. And so that's why I strongly encourage you to come and talk to us about how best to do that. It can involve either things like mechanical lysis through sonication or pulverizing. It can be through chemical denaturation using detergents. But ultimately what you're aiming to do is to generate a sample that is free of DNA or RNA, other biological molecules that we're not particularly interested in, specifically talking about proteomics, something that is enriched in your proteins of interest. And what we want to do is a protein denaturation. We want to unfold those proteins so that we can go on to subsequent steps. We do that best by using chaotropic agents, things like urea, and that'll really help unfold our proteins. The next step is we want to then, by unfolding the proteins, something that holds them together, even under highly denatured environments, is our secondary structure that we have in biology. And for proteins, that is frequently dominated by disulfide bridges, which you find between cysteine residues. That'll hold it together. We reduce the disulfide bonds through action of things like dithiothretol, TSEP. We have these reducing agents that we do in our lab. And then once you form these free thiols, we actually cap them. We alkylate them so they don't reform and they don't scramble the protein sequence. And so that is something we do. And if you don't perform these steps prior to the next step, you can actually get not great results. The final step then is the enzymatic cleavage, or the digestion going from proteins to peptides. And we can do that by adding a number of different enzymes. And all of you, I'm sure, use various enzymes in whatever workflows that you're using within your own labs. And the idea is that you're going to produce peptides of various lengths. Some of them are going to be unique for your protein of interest and will help with the confident identification and quantification of your protein. And so some examples are here, things like aspen, gluci, lysi. These all give an indication of where in a protein they will specifically cleave. But the one that is preferred predominantly in modern proteomics is trypsin. And we do that because trypsin has a very specific cleavage sites. I'm sure many of you know this. It is C-terminal to lysine and arginine residues within your protein. I've just drawn up an example protein sequence, totally made up. And I've highlighted in red those lysine and arginine residues spread throughout the protein. And so if we were to digest this particular protein with lysine, what you end up with is a bunch of peptides that terminate in lysine and arginine. Lysine and arginine are the most gas phase basic amino acids. And that means that when we transfer them to the gas phase, the ions, using an ion source, they will actually hold a positive charge, which is exactly what we need. As I said, it's mass spectrometry is a technique that we require your analytes to be ionized. So we're already putting ourselves in good stead for generating peptides that are going to be positively charged. The next section is now we have our positively charged peptides, is we want to use an orthogonal technique before the mass spectrometer to separate them. And we do that using nano-liquid chromatography. And it's called nano because we're using flow rates in hundreds of nanoliters per minute. I'm sure many of you have done HPLC or size exclusion. You're probably working in the mil per minute flow rate. This is nanoliter per minute. This is very, very sort of slow, great separation for getting peptides separated in LC. We load your peptides onto a C18 column, and then we're applying an elution gradient to separate the peptides from the LC into the mass spectrometer. And we do that by increasing the percentage of organic solvent in our mobile phase. We use acetonitrile. And so at the beginning of the gradient, we're going to see your most hydrophilic peptides, and we've optimized our conditions so we aren't losing sort of highly hydrophilic peptides. And then as we elute across our gradient, your peptides are eluting. We're giving time for the mass spectrometer to isolate, fragment your peptides. And so towards the end of the gradient, you're going to get hydrophobic peptides as well. So it's not sort of preferential for any particular type of peptide from your global proteomics set. Once we've separated the peptides by nano-LC, we need to transfer them from the LC column into the mass spectrometer. And we do that using a technique called electrospray ionization. And it's just an incredible technique. So up until basically the early 1990s, this wasn't a technique that it hadn't been sort of pioneered and discovered in a good way. Previous mass spectrometry work done before that was done on analytes and compounds that were easily ionizable or easily fragmented. And so work that was done by John Fenn at Yale allowed them to figure out transferring macromolecules, biomolecules like proteins and peptides from the solution phase into the gas phase. And so by doing that, it minimized any fragmentation. You were getting intact transfer of peptides and proteins into the gas phase. John Fenn and some others won the 2002 Nobel Prize in Chemistry for making elephants fly, which, depending on my mood, is either that sort of concept is either very interesting and romantic or terrifying to have elephants with wings. And I'd strongly recommend, if you get a chance, his Nobel lecture of electrospray wings for molecular elephants. It's actually really beautiful. It's a very nice lecture. There are a bunch of other ionization methods available depending on the type of technique you're using. Some you may be familiar with are things like MALDI, which is where you fire a laser at your analytes of interest. ICP, which is inductively coupled plasma. That's done a lot for people that are doing maybe elemental mass spectrometry. And then DESI is a type of technique that's used quite a lot within areas of security, where you can actually apply a spray to people's fingertips and determine drug content that are coming. The sort of drugs, class A drugs, can be ionized with fingerprints. These are fascinating areas as well. So don't go down that rabbit hole of Googling these things and just losing hours to looking at these. The next step is, once we have our peptides ionized and into the mass spectrometer, how are we detecting them and ultimately fragmenting them? So when we transfer it, so this little sort of string of beads that you can see here is meant to represent an intact peptide. It's transferred into our mass spectrometer and we collect an MS spectrum, or MS1 level. And that is where we're going to generate an M/Z value that corresponds to the intact peptide. So that is a great way for us not only to use the liquid chromatography, each peptide is going to have a representative retention time, but it's also going to have an M/Z value. And we're able to accurately and precisely measure M/Z values to 3 or 4 decimal places. So that helps to distinguish different species in the gas phase. What we can then do is we can tell the mass spectrometer to isolate and fragment that individual intact peptide to form what is called an MS-MS spectrum, or MS2 level. And so what you can see here is the string of beads on the left-hand side, and then on the right-hand side for each loss you're seeing here, this peak will be for the intact peptide losing additionally one amino acid as you go across. So you actually build up this, you can actually do a peptide ladder to determine the sequence of your of the amino acids for your peptides. So again, this is simplifying it in a way that just shows and represents what we're doing. This then happens hundreds, if not thousands of times within very short timescales in the mass spectrometer, then it sort of amplified across all of your sequence and all of your peptides. And then once you've actually decided upon how you're going to analyze your samples, we have a huge array. Well, I've shown here 5 examples, but there is really quite a huge array of different options for how best to acquire the data within the mass spectrometer. We will help direct and decide, or will support which way you want to do this, but ultimately it falls under 2 main flavors: untargeted and targeted. So for a lot of the bottom-up research work that we do, you're either trying to explore or have a discovery for which peptides and proteins are present in your sample. And there's 2 main flavors of that. One is data-dependent acquisition, or DDA. The other is data-independent acquisition, or DIA. And I'll talk about those next. Away from bottom-up proteomics, like for our histone work or for our dedicated analyte work, we use what's called targeted proteomics. And as you can see, there's a lot of acronyms in generally all of omics. But for targeted work, it can be single reaction monitoring, where you're looking for one very specific analyte within a soup of all the other ones. Multiple reaction monitoring, which allows you to look at many specific analytes within a sample. And that's actually how we do our histone analysis. And then a very cool technique that I used to use a lot is parallel reaction monitoring, which we can do as a targeted approach on our Orbitrap instruments. And this is another area that, if for any of you that wish to explore some more PRM work, come and talk to me as well. That's not something that's been done historically very much at Northwestern, not for any particular reason, but it is something I would love to start offering more for some dedicated projects. So as I said, DIA and DDA is for bottom-up or discovery proteomics. And then for targeted, it's going to be specific analytes or your histone assay or the histone panel we do. So let me talk a little bit about DDA. So on the top of this slide, what we're showing is that at the MS1 level, when the instrument is working just with the intact peptides before we've done any fragmentation, the precursors, another word for the intact peptides, if they are above a specified intensity, we can make that intensity quite small. Anything above that dotted red line will be selected, and then the instrument will create a fragmentation spectrum for that. And so this is an excellent way for getting very high quality MS-MS spectra. And ultimately, what you get is you get quite simple or clear MS2 spectra. The data analysis is relatively easy. A lot of the pipelines that we have are set up to do this. It's very straightforward to do. And so it's called data dependent because the acquisition of this MS2 level data is dependent on the MS1 information. But some of you might realize that, what about those gray bars below the dotted line? Are they not being isolated and fragmented? The answer is most likely not. And so that is where, if you have very low abundance peptides that are eluting at the same time as some very high abundant one, you can get MS1 level data, but not MS2. And we call that missing values or missing data. And so that is one of the areas that the community has really been trying to improve and to come up with some very smart ways of doing it. So we have done things whereby we lower that dotted red.
I'm going to mute.
Could everyone just mute if you're on Zoom?
So I think we should reach out to this.
We'll carry on. And so we do something called wide window.
In the past, the worry is that a few times we had in day biobank, we needed to access some sample, and they were not always available to get us the sample to do our experiment, and that was a concern. Would you have access to it?
Right.
That's important.
It's probably
I thought that was someone wanting to ask something. I was like, huh? So yeah, this type of technology is improving I would say every 6 months, we're getting very interesting new ways of improving those missing values. And for any of you who've already done some proteomics with some global or complex proteomics and have seen the challenges with missing values within particularly interesting data sets, we're addressing that with acquisition using wide window acquisition. The other reason that because this produces simpler MS2 and sort of easier data analysis, I'm going to go on to talk about something that historically has been more complex or more challenging. So for data independent acquisition, what you're doing instead is that you're selecting an M/Z range. So in this top example, the blue, the orange, the purple, and the green are, let's say, 100 M/Z windows. Blue might be 300 to 400 M/Z, the orange is 400 to 500, etc., and you go up. And what the instrument does in that blue range, it's selecting everything in there, all the precursors, all the intact peptides, fragmenting all of them, and forming an MS2 spectrum, which is really quite challenging to analyze. It's very messy, it's very noisy. We refer to it as having chimeric spectra in it. And so for quite a long time, so DIA has been around for quite a while, but the application of it, especially within a core environment, it has been very hard. And so with the arrival of Simon, it's been great that we've started to jump on this. And we're actually collecting some current projects we're collecting in our DDA workflow as normal, but we're collecting parallel data in DIA. And so in 2025, I hope to be able to offer any of you the option to do DDA and or DIA. And so ultimately what you're getting, acquisition of MS2 data is independent of the MS1 information. Okay. A couple of examples for the type of work that we do for bottom-up are shown here. I've given 4 very broad examples because the list is relatively exhaustive. A common one is protein identification from SDS page gels. This is where proteomics started, kind of in the 90s and the early 2000s, and where many of you may recognize it from. And it's where you would run an SDS page gel. You may have one band or many bands. People sometimes, as part of a purification process, have an unknown protein being produced, some sort of contaminant, some sort of non-specific protein, and they want to know what it is. And they give us that gel band, and we're able to identify what it is. And it's so often something like a chaperone protein or a heat shock protein, a folding protein that is part of the process, and it really helps alleviate people's concerns about what it is. And so very straightforward. You're able to give us SDS pH gels that have been stained with Kumasi. We will excise that stained gel band. We'll go through all the steps I described earlier about digesting it, getting the peptides, and give you a very clear answer. This is very quick and easy. The next example on the lower left is label-free quantification, so LFQ. And this is where a lot of people come to us. They say, I want to know within the system I'm interested in, what proteins are present and how their abundance is changing between different conditions. And so that is an excellent way for us to use our DDA, but going forward to using our DIA is going to be great. And so that is a label-free quantification process. You're going to identify 6,000 to 7,000 proteins within your samples. For that, you're going to need to submit replicates so that we can do some very nice, robust statistics on it. And again, Simon has been producing some nice reports that help clarify what it is you're seeing. In the top right, something that a lot of people want to do is protein-protein interactions. Now, maybe you have a particular protein, a bait protein, and you want to know what are its interactors under a particular set of conditions. And so what I would encourage people to do is to use maybe affinity purification. If you have a good antibody that recognizes your bait protein, you could covalently attach your antibody to magnetic beads. You pull out your bait protein and its sort of associated interactors. And then you can give us those. We will do an on magnetic bead digest to give you the different proteins. But for that, obviously, you need good negative control. And I say magnetic beads because for a long time, we would encourage people to use agarose or sapphirose beads, but these are porous, as you know. Small proteins, especially ribosomal proteins, love to get stuck inside those sapphirose beads. And no matter what you do in terms of washing or sort of various different controls or replicates, you see this background constantly of these proteins. So I would encourage you to consider moving. So that's why I, again, encourage you to come talk to us beforehand, just knowing what are some of the common pitfalls. Another really common area that we're working with is in proximity labeling. So people are maybe have biotinylated or GFP type proteins, and they want to see in a particular environment what is being what are the local interactors, and that's done by things like BioID, TurboID. We can do all of those, and we have great protocols, methodologies to handle them. And then finally, something we started to do an awful lot more of recently is post-translation modifications. We have great protocols for phosphoproteomics, and you have to actually enrich for the phosphorylated peptides within the system. There are some very nice techniques that I've used for years. One of them is called SMOKE, which is sequential metal oxide affinity chromatography. And that's where we pass peptides that are phosphorylated across an iron-containing column and a titanium dioxide column. That really enriches mono and multiply phosphorylated peptides. We have some very nice workflows if you're interested in acetylated, methylated, or ubiquitinated proteins. And this involves using antibody enrichment techniques to enrich for peptides that have these modifications on them. These are very low abundance within a biological system, so you sometimes have to start with a relatively high amount of material. And that, again, can be the difference between somebody's working in cell culture on the cell line, which is relatively easy to work on, versus something that's trickier. And having those conversations with us early in the process will help set you and your PI's expectations of what we can do. But no, we'll always try to do it. Yes, simulation is another great one, and it works sort of hand in hand with ubiquitination as well. Okay, how are we doing for time? What time did we start? Right after, okay. Another cool example I wanted to show you, another buzzword that comes up is TMT, or tandem mass tag. This is a type of proteomics technique which it's quantitative, so it's both identification and quantification. And it's also what we refer to as a multiplexing approach. So it means that you're able to take a number of samples, analyze them all together within the mass spectrometer, which reduces any sort of biases, but it increases your protein quantification accuracy and precision. This technique, let me give you an example. So if you have, let's say, 5 or 6 cell cultures, you have a control sample and 5 experimental conditions. That can be anything. It might be a particular drug, and each of the 5 experimental conditions perhaps are an increasing concentration of your drug. First step would be you lyse your cells, you collect your proteins, and you triptychally digest them to form peptides. So you would do that identically on each of your 6 conditions. Then the next step is you would label, well, we would label them using these TMT labels. What a TMT label is that each label has a distinct detectable reporter ion mass. We're basically barcoding the samples in a really great way. So in this example, what we're doing is taking the first set of peptides from the control experiment, and we're labeling them with the blue TMT label. This label has a specific mass. We're taking all of the peptides in experiment number 1. We're labeling them with the red mass. We continue this on. And I do it in terms of color, because right now, when I first started, there was only 6 options, TMT 6-plex. Now I think Steve Geegy and the guys in Harvard are up to 32. So I think the rainbow of colors I would have to come up with would be quite large. But this is a really great technique for getting highly quantitative experiment for your protein. For your proteins across a number of experiments. And so once you have labeled each one of your samples with its distinct TMT label, the next step is that you actually pool all of your samples together into one, because now you've barcoded each of them. You have a way of determining the identity and the quantity of each. So what we then do is we pool them all, we analyze them using, actually that instrument I talked about earlier, the one in the middle, the Tribrid Ascend, is perfect for this type of analysis. And what you're doing is you collect MS1 level where actually the peptides in each of the conditions are indistinguishable. And it's at the MS2 level that we now, when we fragment the peptides, we not only get fragmentation and sequence information, we get these reporter ions, which we can use as a quantitative readout. And so this example is sort of the 6 readouts for each of the for each peptide within the sample. So this is an incredibly complex data set that we then use excellent bioinformatic resources to tease apart. And again, TMT is something that's being done a little bit historically, but I want, and any of you want to do TMT, come and talk to me. It's something I did a lot at the Francis Crick in London. Obviously, we more than happily can give you the raw data. Should you want to do your own data analysis, we will do all of the data analysis for you, or we can show you how to do your own data analysis. That's what we're here for, here to educate and help train the next generation of scientists. The way that we then take the data, we use informatics to do comparisons, matching, and scoring for all of that data against an in silico digested protein database using a FASTA proteome from a website such as Uniprot. So we have very nicely curated proteomes for the vast majority of species that people are looking at. And then, as I already mentioned, the identification is usually performed at the MSMS level, that fragmented level, and the quantification is done at the MS1 level. As I already mentioned, oh, Simon's cut off at the top. Simon is our excellent new bioinformatics researcher. He has some great access to good knowledge R scripts. If you have anything on GitHub or anything you want to work with, we have all of that. We have a bunch of amazing softwares. Again, just to say them out loud, so if you recognize anything, things like MaxQuant, Proteome Discover, Scaffold, FragPipe, Diane, which is DIA neural network, Skyline, and Spectrometry. We have access, we have all of these. The ones, some of them are free, some of them are licensed. We have them here to support you guys. We are going to give you back really clear results for ID and quantification. Simon and I are developing tailored R scripts for particular projects, and we will work with you if you want to customize something or you want something very specific that's going to be useful for you or the research group you're in, or for other people. Please come and talk to us. Briefly, I'm going to talk about histone work. So for this is where we use our triple quad mass spectrometers. We do quantification of histone marks. I'm sure histone marks may be familiar to some of you. That's specific chemical modifications on histone proteins associated with different states of gene transcription. They're predominantly on lysine residues, things like mono, di, and trimethylations and acetylations, but there's a vast array of the histone code. So come and talk to us about what you want to do in that. We have very specific and tailored approaches for producing samples for histone pellets that use downstream. It's not the same as our bottom-up work. We actually block unmodified lysines with a propionylation agent. And we do a trypsin digest, so it is a little similar to bottom-up work, but again, we're using targeted triple quad mass spectrometry analysis and using an MRM targeted approach. So we have a very dedicated list of precursors and product ions. So we have a list at the minute of more than 100 histone marks. We're always developing that. So for any of you who are working on histones, come and talk to us. Some of the marks you want to do, we may not currently do. We want to develop and grow those. We need your help to do that. Example of it is maybe this AKA KTR peptide. We have very dedicated, the precursor ion is going to be at this particular mass. It comes out, it elutes from the chromatography at this specific time. We isolate a very specific M over Z, and then we only monitor a particular number of ions. So it is incredibly dedicated and specific for the ion that you want to look at. We have knowledge within histones of a particular peptide, how they elute from the chromatography based on their modifications. That is quite simplistic. Of course, with combinatorial approaches, combinatorial masses, you get a huge array of potential peptide combinations. But as I said, we want to develop this technique even more, and we're going to need people like you, and at Northwestern, we need your help to do this. I'm going to briefly skip over top-down proteomics. It's done in collaboration with Neil Kelleher's lab up in Evanston. It's where you're looking at intact proteins, PTMs, proteoforms, antibodies, those sorts of things. If any of that rings a bell to you, come and talk to me. I wanted to give one very cool example of something outside of the research we do at at Northwestern, which is using mass spectrometry to detect biomarkers. Ultimately, I think a lot of you know what biomarkers are. It's going to be something that gives a conclusive early stage detection of a particular disease state. One in particular, I know a lot of people are working on Parkinson's disease. And so is there a way to diagnose Parkinson's non-invasively and with confidence? As I mentioned before, I did my PhD in Scotland. This is Joy Milne. She's a retired nurse from Scotland, and she could actually smell Parkinson's on her husband. It is an incredible story. I encourage you to look into it. And effectively, the way to think of it is a lot of people think of mass spectrometers as noses. And so she teamed up with Purdy Barrett, the person I did my PhD for. Purdy and Joy worked together and used gas chromatography mass spec to identify a particular biomarker in sebum, which is a secreted oil-like substance. Google Joy Milne. Purdy has a TEDx talk from Palo Alto. This is really magical, inspirational stuff and reminds us possibly of why we do the type of research we do. And I just think it's a really cool story. Okay, I think I've shown you some of the approaches that we have available here at Northwestern. We have Orbitrap instruments, triple quad instruments. We don't have any TOF or timsTOF instruments. So if you're interested in that, please come and talk to me. We're here to support you. We're an excellent complementary technique for any of you that haven't done mass spec or proteomics before. That's our email address. That email is proteomics@northwestern.edu. That comes to me. Email that or email me, Peter Faull. We're based next door in Olsen up on the 8th floor. You're all welcome anytime to come and visit. But email, come and talk to us about your research. I think, yeah, that's everything. Thank you very much.